Somewhere inside every cell in your body, there is a library, 3 billion letters long, written in a code so old it predates language, predates fire, predates the first heartbeat of the first creature that ever crawled onto dry land. For 4 billion years, that library edited itself only by accident. A mutation here, a survival there, a slow, blind, patient process we call evolution, working without a plan, without an author, without a single moment of intention. And then, in a lab in California in 2012, 2 scientists discovered something that had never existed before in the entire history of life on Earth. A pair of molecular scissors precise enough to open that library, find a single misspelled word among 3 billion and rewrite it. They called it CRISPR. And the moment they published their findings, the rules that had governed life since its very beginning, quietly, irreversibly changed. To understand how strange this moment truly is, you have to understand what came before it. For most of human history, we could only watch evolution happen to us. We buried children who inherited diseases we didn't understand and couldn't name. We watched genetic conditions pass silently from parent to child, generation after generation, like a curse with no origin and no cure. Doctors could treat symptoms, manage pain, extend life by years or sometimes decades, but the code itself, the actual instructions written into every cell, remained untouchable. It was the one part of a person that medicine could never reach, and then it could. The technology that made this possible has a strange and beautiful origin story, one that has nothing to do with human medicine at all. Scientists studying ordinary bacteria noticed something odd. These single-celled organisms, constantly under attack from viruses, had developed their own immune system, a way of literally cutting viral DNA out of existence and keeping a genetic mugshot of every invader they'd ever survived. It was, in essence, evolution's own editing tool, hiding in plain sight inside the simplest life forms on the planet. Jennifer Doudna and Emmanuel Charpentier realized that if you could reprogram this bacterial defense system, you could point not at viruses, but at any DNA sequence you chose, including our own. In 2020, they won the Nobel Prize for that discovery. But the real story wasn't in the award ceremony. It was in what happened next, in laboratories and hospitals and courtrooms around the world, as humanity quietly began deciding what kind of species it wanted to become. The first place this new power showed its face wasn't in some dramatic redesign of human beings. It was in the quiet, desperate hope of families who had run out of options. Consider sickle cell disease, a condition that twists red blood cells into rigid crescents, cells that clog blood vessels and cause pain so severe that patients describe it as being crushed from the inside. For over a century, the only real treatments were blood transfusions and pain management, a lifetime of managing suffering rather than ending it. Then, clinical trials that read like science fiction, doctors extracted patients' own bone marrow cells, used CRISPR to switch a single genetic switch back on, a switch that normally shuts off after infancy, and returned the edited cells to the body. Patients who had spent decades in and out of emergency rooms went months, then years, without a single crisis. In late 2023, this became the first CRISPR-based therapy approved for human use, a moment that historians will likely mark the way they mark the discovery of penicillin, not because it cured everything, but because it proved the door could be opened at all. But here is where the story stops being simple and starts becoming the kind of question that keeps ethicists, scientists, and parents awake at night. Editing a patient's own cells to treat a disease they already have is one thing. It affects only them. The change dies when they do. But there is another kind of edit, one that doesn't stay contained to a single lifetime. It happens in embryos, in eggs, in sperm, in the earliest possible moment of a human life, before a single cell has divided. Edit there, and you are not just treating a patient. You are rewriting the instructions that will be passed down to their children and their children's children, for as long as that family line continues. You are, for the first time in the story of life on Earth, making a decision about evolution on behalf of people who don't exist yet. In November 2018, a scientist named He Jiankui announced that he had done exactly that. Working in secrecy in China, he had used CRISPR on human embryos to disable a gene called CCR5, attempting to make the resulting children resistant to HIV. Twin girls were born. The scientific world did not celebrate. It recoiled. Within months, He Jiankui was stripped of his positions, condemned by researchers worldwide, and eventually sentenced to prison for illegal medical practice, not because the science was fantasy, it worked more or less, but because he had crossed a line that the Global L scientific community had spent years agreeing, informally and formally, not to cross. Editing the human germline, The code passed to future generations without anywhere near enough understanding of the consequences, without transparent oversight, and without the informed consent of children who would carry those edits for the rest of their lives and pass them on to lives not yet imagined. The fear wasn't abstract. Scientists studying the CCR5 edit later found troubling evidence that the same mutation, while offering some protection against HIV, appeared linked to a measurably higher risk of early death from other causes, including flu. In trying to remove one vulnerability, the edit may have quietly introduced another, a reminder that our genome isn't a simple list of independent switches we can flip in isolation. It's an intricate, densely interconnected system, 4 billion years in the making, where a single gene often does far more than the one job we notice. Evolution didn't design us with a user manual. It designed us through trial and error on a scale of billions of years and trillions of lives. We're now trying to read that manual in real time, with tools that let us make changes faster than we can fully understand their consequences. And yet, the door is open. It cannot be quietly closed again, which means the real question facing this generation isn't whether gene editing will shape human beings. It already has. The question is, who gets to decide on how far it goes and where the line sits between healing and enhancing? Almost everyone agrees that curing a child of a fatal genetic disease is an act of mercy. But what about editing embryos to reduce the risk of conditions like Alzheimer's, decades before symptoms could ever appear? What about height? What about intelligence, if such a thing could even be isolated to a handful of genes, which most scientists doubt it ever will be? Each of these questions sits on a spectrum, and there is no obvious point on that spectrum where a bright red line simply appears. That ambiguity is precisely what makes this moment in history so fragile and so important. There's a deeper unease underneath all of this, one that has nothing to do with the science and everything to do with fairness. Advanced genetic therapies are extraordinarily expensive. The sickle cell? Treatment approved in 2023 carries a price tag in the millions of dollars per patient. If the ability to edit out disease, and eventually perhaps to edit an advantage, remains available only to those who can afford it, we risk building something history has never quite seen before, a genetic class divide written not into bank accounts or zip codes, but into DNA itself, inherited and compounding across generations. It's a quiet kind of inequality, one that wouldn't announce itself with a headline. It would simply show up decades from now in life expectancy statistics, in disease rates, in outcomes so gradually diverging that by the time anyone noticed the gap, it would already be generations wide. And still, alongside every one of these fears, there is something undeniably hopeful happening. In laboratories right now, researchers are using these same tools to fight cancers that were considered untreatable a decade ago, training a patient's own immune cells to recognize and destroy tumors with a precision chemotherapy could never achieve. They're developing gene therapies for inherited blindness, restoring sight to people who had never seen a sunrise. They're working on treatments for muscular dystrophy, for cystic fibrosis, for a long list of conditions that have caused immeasurable suffering for as long as human beings have kept written records. For every anxious headline about designer babies, there are 100 quiet, unglamorous experiments aimed simply at reducing the amount of pain in the world. That's the part of this story that rarely makes it into the dramatic documentaries and the panicked op-eds. Most of the people doing this work aren't trying to redesign humanity. They're trying to stop a child from dying of a disease their grandparents also died of, and their grandparents before them. So where does this leave us, standing at what may be the strangest crossroads our species has ever faced? For the first time since the very first cell divided in some ancient ocean, a species on Earth has the tools to read its own instructions, and the growing ability to rewrite them. We didn't earn this power through some slow evolutionary process. We built it in the span of a single human lifetime in labs, powered by curiosity, ambition, and yes, our own mortality. What we do with it now will not be decided in a single dramatic moment. It will be decided the way most of history is actually made, in thousands of small decisions, in hospital ethics committees and international treaties, in courtrooms, and kitchen table conversations, between parents faced with impossible choices in the quiet. Persistence of scientists who believe that suffering isn't something we simply have to accept. Evolution spent 4 billion years writing the human story one accident at a time. We are now the first generation in that story who gets to hold the pen. What we choose to write next won't just describe who we are. It will define who our descendants become for every generation that follows, for as long as there are humans left to read the code we leave behind.